This video is about somatosensory cortex. We know that our brain, human cerebral cortex in particular, is divided into about 50 different areas with the help of a map called Brodmann areas based on histological structural differences. Brodmann areas with the help of histological structural differences. This map is important because virtually all neurophysiologists and neurologists use it to refer by number to many of the different functional areas of the human brain, and it is to a greater extent related to the histological differences. Histological structural differences. Okay, histological structural differences. Large central fissures, also called central sulcus, that extends horizontally across the brain. In general, sensory signals from all modalities of sensation terminate in the cerebral cortex immediately posterior to the central fissure. Generally, the anterior half of the parietal lobe is concerned almost entirely with reception and interpretation of somatosensory signals, but the posterior half of the parietal lobe provides still higher levels of interpretation. Visual signals terminate in the occipital lobe and auditory signal terminate in the temporal lobe. So conversely, the portion of the cerebral cortex anterior to the cerebral fissures and constituting the posterior half of the frontal part is called the motor cortex and is devoted almost entirely to control the muscle contractions and body movements. A major share of this motor control is a response in response to somatosensory signals perceived from sensory receptor portions of the cortex, which keep the motor cortex informed at each instant about the positions and motions of the different body parts. And somatosensory areas 1 and 2. Two separate sensory areas in the anterior parietal lobe called somatosensory area 1 and somatosensory area 2. The reason for this division into two areas is that distinct and separate spatial orientation of the different parts of the body is found in each of these two areas. However, somatosensory area 1 is much more extensive and so much more important than somatosensory area 2. That in particular usage term, somatosensory cortex almost always means area 1. Next is about somatosensory area 1 has a high degree of localization of the different parts of the body. Localization is poor in somatosensory area 2, although roughly the face is represented anteriorly and the arms centrally and the legs posteriorly. Much less is known about the function of somatosensory area 2 and its localization is, to a great extent low. So it is known that signals enter this area from the brainstem transmitted upward from both sides of the body. In addition, many signals come secondarily from somatosensory area 1 as well as from other sensory areas in the brain even before the visual and auditory areas. Projections from somatosensory area 1 are required for function of somatosensory area 2. Projections from somatosensory area 1 is required for function of somatosensory area 2. However, removal of parts of somatosensory area 2 has no apparent effect on the response of the neurons in somatosensory area 1. Thus, much of what we know about somatic sensation appear to be explained by the function of somatosensory area 1. Spatial orientation of signals from different parts of the body in somatosensory area 1. Somatosensory area 1 lies immediately behind the central fissure located in the postcentral gyrus of the human cerebral cortex. Brodmann area 3, 1 and 2. Postcentral gyrus demonstrating representations of the different parts of body in separate regions of somatosensory area is very important. This representation is called homunculus. Each lateral side of the cortex receives sensory information about extensively from the opposite side of the body. Some areas of the body are represented by large areas in the somatic cortex, the lips in the greatest of all, followed by the face and thumb, whereas the trunk and lower parts of the body are represented by relatively small areas. The sizes of these areas are directly proportional to the number of specialized sensory receptors. The sizes of these areas are directly proportional to the number of specialized sensory receptors in each receptive peripheral area of the body. A great number of specialized nerve endings are found in lips and thumb, whereas only a few are present in the skin of the body trunk. Layers of the somatosensory cortex and their function. The cerebral cortex contains 6 layers of neurons, beginning with layer 1 next to the brain surface and extending progressively deeper to layer 4. And as would be expected, the neurons in each layer perform functions different from those in other layers. Some of these functions are as follows. The incoming sensory signal excites neuronal layer 4 first, and the signal then spreads towards the surface of the cortex and also towards deeper layers. Layer 1 and 2 receive diffuse non-specific input signals from lower brain centers that facilitate specific regions of the cortex. Neurons in layer 2 and 3 send axons to related portions of the cerebral cortex on the opposite sides of the brain through the corpus callosum. The neurons in layer 4 and 5 and 6 send axons to the deeper parts of the central nervous system. Those in the layer 5 are generally larger and project to more distant areas, such as to the basal ganglia, brainstem, and spinal cord, where they control signal transmission. From layer 6, especially large number of axons extend to the thalamus, providing signals from the cerebral cortex that interact with and help to control the excitatory levels of incoming sensory signals entering the thalamus. So different Brodmann areas is there in the human cerebral cortex we mentioned earlier. And homunculus is a representation which contain portions like intra-abdominal, pharynx, tongue, teeth, gums, and jaw, lower tip, lips, upper lip, face, nose, eye, thumb, index finger, middle finger, ring finger, little finger, hand, wrist, forearm, elbow, arm, shoulder, head, neck, then trunk, hip, leg, foot, toes, genitals, etc. The sizes of these areas are directly proportional to the number of specialized sensory receptors in them. Functions of somatosensory area 1. Widespread bilateral accession of somatosensory area 1 causes loss of following types of sensory judgment. A person is unable to localize discretely the different sensations in different parts of the body. However, he or she can locate these sensations crudely, such as the particular hand or a major level of body trunk or the legs. Thus, it is clear that the brainstem, thalamus, or parts of cerebral cortex not normally considered to be concerned with somatic sensation can perform some degree of localization. The person is unable to judge critical degree of pressure against the body. The person is unable to judge the weights of objects. The person is unable to judge shapes or forms of objects. This condition is called asterognosis. The person is unable to judge texture of materials because this type of judgment depends on highly cortical sensation caused by movement of the fingers over the surface of to be judged. This list has been said about the loss of pain and temperature sense. In the sensor specific absence of only somatosensory area 1, appreciation of the sensory modalities are still preserved in both quality and intensity, but the sensations are poorly localized, indicating that pain and temperature localization depend greatly on the topographical map of the body in somatosensory area 1 to localize the source. Somatosensory association areas. Brodmann areas 5 and 7 of the cerebral cortex, located in the parietal cortex behind the somatosensory area 1, play important roles in deciphering deeper meanings of the sensory information in the somatosensory areas. Therefore, these areas are called somatosensory association areas. Which areas? Area 5 and 7. Electrical stimulation of a somatosensory association area can occasionally cause an awake person to experience a complex body sensation, sometimes even the feeling of an object such as a knife or a ball. Therefore, it seems clear that the somatosensory association area combines information arriving from multiple points in the primary somatosensory area to decipher its meaning. This occurrence also fits with the anatomical arrangement of the neuronal tracts that enter the somatosensory association area because it receives signals from somatosensory area 1 and the ventrobasal nuclei of the thalamus, other areas of the thalamus, and the visual cortex and the auditory cortex. An effect of removing the somatosensory association area, amorphosynthesis. When the somatosensory association area is removed on one side of the brain, the person loses the ability to recognize complex objects and complex forms felt on the opposite side of the body. In addition, he or she loses most of the sense of form of his or her own body or body parts of opposite side. In fact, the person is mainly oblivious to opposite side of the body. That is, forgets that it is there. This condition is called neglect. Therefore, the person also often forgets to use the other side of motor functions as well. Likewise, the feeling objects, the person tends to recognize only one side of the object and forgets that the other side even exists. Or this complex sensory deficit is called amorphosynthesis. The contralateral loss of ability to recognize objects by touch without usual input is called asterognosis. This in turn requires a capacity for two-point discrimination, as described later. So, overall characteristics of signal transmission and analysis in the dorsal column, medial lemniscal cortical system. Basic neuronal circuit in the dorsal column, medial lemniscal system. The lower part shows the basic organization of the neuronal circuit of the spinal cord dorsal column pathway, demonstrating that at each synaptic stage, divergence occurs. The upper curves of the figure show that cortical neurons that discharge into the greatest extent than those in the central part of the cortical field for each respective receptor. Thus, a weaker stimulus causes only the central most neurons to fire. A stronger stimulus causes still more neurons to fire, but those in the center discharge at a considerably more rapid than do those farther away from the center. And two-point discrimination, a method frequently used to test tactile discrimination is to determine a person's so-called point-to-point discriminatory ability. In this test, two needles are pressed slightly against the skin at the same time, and the person determines whether 1 point or 2 points of the stimulus are felt or 1 point. On the tips of the fingers, the person can normally distinguish two separate points even when needles are close together as 1 to 2 mm. However, if on the person's back, the needles usually must be enough far apart as 30 to 7 mm before two separate points can be detected. The reason for this difference is the different numbers of specialized tactile receptors in the two areas. Mechanism by which dorsal column pathway as well as other sensory pathways transmit two-point discrimination information. Two adjacent points on the skin that are strongly stimulated as well as the areas of the somatosensory cortex that are excited by signals from the two simultaneous stimulated points. The resultant zone of excitation has two separate peaks. These two peaks, separated by valley, allow the sensory cortex to detect the presence of two simultaneous points rather than a single point. The capability of sensorium to distinguish this presence of 2 points of stimulation is strongly influenced by another mechanism called the lateral inhibition, as explained in the next section. Effect of lateral inhibition, also called surround inhibition, to increase the degree of contrast in the perceived spatial pattern. Every sensory pathway, when excited, gives rise simultaneously to lateral inhibitory signals. These inhibitory signals spread to the sites of the excitatory signal and inhibit adjacent neurons. For instance, consider an excited neuron in a dorsal column nucleus. Aside from the central excitatory signal, short lateral pathway transmit inhibitory signals to the surrounding neurons. That is, these signals pass through the additional interneurons that secrete an inhibitory neurotransmitter or inhibitory transmitter. The importance of lateral inhibition that it blocks several spread of the excitatory signals and therefore increase in degree of contrast in the sensory pattern perceived in the cerebral cortex. In the case of dorsal column system, lateral inhibitory signals occur at each synaptic level. For instance, the dorsal column nuclei of the medulla and the ventrobasal nuclei of the thalamus and the cortex itself. In each of these levels, the lateral inhibition helps to block lateral spread of the excitatory signal. As a result, the peaks of excitation stand out and much of the surrounding diffuse stimulation is blocked. The effect is demonstrated by a curve. Then transmission of rapidly changing and repetitive sensations. The dorsal column system is also particularly important in appraising the sensorium of rapidly changing peripheral conditions. Based on recorded action potentials, the system can recognize changing stimuli that occur as little as 1 by 400 of a second. Then vibratory sensation. Vibratory. Vibratory signals are rapidly repetitive and can be detected as vibration up to 700 cycles per second. The higher frequency vibration signals originate from the Pacinian corpuscles in the skin, deeper in tissue, but lower frequency signals can originate from Meissner's corpuscles as well. These signals are transmitted only in the dorsal column pathway. For this reason, application of vibration to different peripheral part of the body is an important tool used by neurologists for testing functional integrity of the dorsal column.